Immune cells operate by many different ways and are essential in health and disease. When immune cells get trained to find foreign peptides from infections or from cancers, antigen-presenting cells train T cells in what's known as the immune synapse. The immune synapse forms between these two cell types for them to enable communication around the CD3 TCR complex. In this complex, there are many immuno-oncology drug targets. The action of CAR T cells on cancer cells also involve reorganization of the surface protein of the CAR T cells. And some CAR T companies are even engineering this spatial organization to make the CAR Ts more efficient. When T cells and other immune cells wander into an infected site or a tumor, they need to form a front and a back end in order to move and to transmigrate between endothelial cell layers. This is the most important part of clearing out a tumor in immuno-oncology. And the front and the back end formation, of course, involves multiple reorganizations of the immune cell surface proteins. When antibody-based drugs bind to cancer cells, there are instances where they reorganize the surface protein of that cancer cell. So when rituximab, for example, binds CD20 on cancer cells, it pulls all the CD20 to one end, clustering the antibodies, which triggers NK-mediated killing of ADCC of these cancer cell lines and makes that much more efficient. So if you look at the tools available to study these processes, which is sometimes referred to as deep phenotyping of immune cells, you can look at high content analyses, which measure multiple proteins on the surface of the cell, but does not localize them. You have CytSeq, AbSeq, CyTOF, and flow cytometry for these measures. But on the other axis, the spatial dimension, the spatial resolution requires a microscope to look where the proteins are in relation to each other. And with the microscope, you can only detect a handful of proteins at the same time. So with molecular pixelation, we are combining the ability to look at many things, also with their spatial localization, only driven by a DNA-based readout. So instead of localizing the proteins with a microscope and light and fluorescence, we are using a purely DNA-based signal, which drives our resolution and multiplexing, throughput, and other capacity measures. Using DNA to solve protein detection problems has historically been very successful. Most notably, the proximity extension assay was developed to drive throughput, sensitivity, and capacity for proteins in blood plasma, which are not localized. Here, 2 DNA tags were bound to antibodies that, upon pairwise recognition, gave a DNA-based signal for reading many proteins simultaneously. In our method, molecular pixelation, we convert the proteins present also into a DNA tag, but this DNA tag also includes the spatial location of proteins. So with Pixelgen, we want to bring the power of DNA-based protein detection to the spatial realm. The molecular pixelation chemistry is only a kit with a few reagents that you mix with your sample in a solution phase, so no microscopes, no photography is needed to make this method work. Typically, you would start the method with your PBMC cells or other immune cell types, and you fix them with PFA to lock the spatial organization into place. Then you bind the cells with antibody oligonucleotide conjugates, or AOCs, that we have optimized and verified for specific binding to these target proteins. So that is in step 1 of the method. It's very similar to standard flow cytometry type staining, where the cells are present in a vessel in solution, and you bind the antibodies, then you wash them, and you add the next reagent. So after binding, in the second step, we perform something that we call pixelation A, and we add our DNA-based molecular pixels. So these are nanometer-sized repeat concatenamers of a certain unique molecular identifier. So each DNA pixel has a unique DNA sequence, which fuses with the sequence on the antibody oligonucleotide that's bound to the cell, forming a colocalization zone, which we call colocalization zone A. And this step is then repeated in pixelation B, but with another set of DNA-based pixels, which effectively colocalizes the colocalization event, forming a complete mesh of 3D-based information of where the proteins are in relationship to each other on a single cell. So the entire method does not need any instrumentation or single-cell compartmentalization, which is common in single-cell research. The pixel structure organizes and figures out what is one cell and what is another cell in the computational data analysis part. So at the end of this reaction, you get a PCR product that you feed into your next generation sequencer, and this sequencer reads about 120,000 reads per single cell, and that is fed into our software, which we call Pixelator, which figures out which cell is which, where the proteins are on the cell, and many other spatial metrics that we will soon get to. So this method requires no optimization and there's no equipment necessary. In the data analysis of this method, each mathematical graph represents one single cell. So in this illustration, the graph data is to the left. The blue spots are proteins present on a certain cell, and to the right you can see how the graph network has formed by starting with the protein ID tag, the zone A tag and the zone B tag, and how that forms a spatial network that can be used and mined with spatial statistic metrics to figure out where the proteins are in relationship to each other without any microscopy. You can also find more information on molecular pixelation in our bioRxives preprint that's available there. This has also been accepted for publication in Nature Methods and should be out online within the first half of 2024. After you run your next generation sequencing reaction, the FASTQ file of the sequence is fed into a software that we've developed here at Pixelgen that we call Pixelator. It's a freeware open source software that you can download from our website that provides you with all the spatial metrics that we've talked about today, as well as a quality control measure.